we're working on the cell. This is a long one. It's going to take us today and tomorrow to get through the cell. My intention was to post the outline of this PowerPoint before class. My computer bogged down and just refused to cooperate, so I will try to get that done before I leave today. Just the outline, not the pictures, not the links, just the outline. Okay, so if you have trouble keeping up with notes, and I do recommend that you take notes, but if you have trouble keeping up, you'll know that I'm going to post at least the broad outline of it at some point. So when we talk about cell biology, I encouraged you yesterday to kind of try to review the cell organelles, because I'm really not going to take class time to go over things like mitochondria and rough endoplasmic reticulum. I'm going to assume you know some of that stuff. My main goal today and tomorrow is to convey the content in a way that lets you make some clinical judgments about it, to make some applications of, if this is the way the cell works, then that explains this disorder or this condition. Does everybody see where we're going with that? Okay, so if you have questions, all you have to do is let me know. So the first thing we're going to look at is an overview of cell function. I snagged this picture off of one of my favorite websites, which is one produced by Harvard. I actually got to meet one of the guys that helped develop these. This is a computer model based on X-ray diffraction images of the actual molecules. So this is what this stuff really looks like. And of course, across the top, can you see the phospholipid bilayer? Is that showing up? Not great, but sort of. And then this right here is a big protein. Do y'all remember that we talked about proteins and how they have to fold? Yeah, okay, so that protein is properly folded. Do you see the little piece of the protein that sits in the membrane? Let me see if I can change my pen color to something more visible. That helps. And do you remember yesterday I held up my model and said if it doesn't fold right, if this little part doesn't fold right, it has the wrong shape. So very often for proteins, what we find, if there's a mutation that prevents them from sitting in the membrane the way they're supposed to, then that protein is basically absent from the cell. And that person will have a deficiency based on the fact that that protein, even if that protein is being made by the cell, if it's made wrong and doesn't park itself up in the membrane, then it's not able to do its job. So how important is it for proteins to fold correctly? Absolutely crucial. They cannot do their job unless they're folded correctly. Okay, so that's something to keep in mind. I don't know if my animation is going to work. I'm going to try and see if it does. If it doesn't work here, I'm going to go to, oh, it looks like it is. Except, of course, no sound because I'm not hooked up to the sound. Can y'all hear that well enough?
What I am going to show you are the astonishing molecular machines that create the living fabric of your body. Now, molecules are really, really tiny. And by tiny, I mean really. They're smaller than the wavelength of light, so we have no way to directly observe them. But through science, we do have a fairly good idea of what's going on down at the molecular scale. So what we can do is actually tell you about the molecules, but we don't really have a direct way of showing you the molecules. One way around this is to draw pictures. And this idea is actually nothing new. Scientists have always created pictures as part of their thinking and discovery process. They draw pictures of what they're observing with their eyes through technology like telescopes and microscopes, and also what they're thinking about in their minds. I picked two well-known examples because they both they're very well known for expressing science through art. And I start with Galileo, who used the world's first telescope to look at the moon, and he transformed our understanding of the moon. The perception of the 17th century was the moon was a perfect heavenly sphere, but what Galileo saw was a rocky, barren world, which he expressed through his watercolor painting.
Okay, that's all fascinating, but we need to get to the molecular biology.
I encountered the artworks of David Goodsell, who was a molecular biologist at the Scripps Institute. And his pictures are all, everything's accurate, it's all to scale.
So what you see in this picture
Mark's work illuminated for me.
What you see in this picture is everything except maybe the water molecules. So that's a cell there. Can y'all tell that that's a cell? That's the bottom right, I'm pretty sure, is a red blood cell. That's hemoglobin molecules in red. And so can you see the phospholipid bilayer kind of curving across? And then everything to the left that's in yellow and green, that's all the plasma. And so you're seeing that plasma is full of stuff. It's mostly proteins in that picture. All right, we unfortunately can't take the class time to view this whole thing, but I'm going to tell you to write down this guy's name, Drew Barry, and Google him. Google his TED Talk.
What the molecular?
Because what you see in this image is actually a still from an animation that shows DNA being replicated. And they have this down to, astonishing accuracy in terms of what it looks like when DNA replicates. And we know this. This is not just an artist's rendering. This is actually based on measurements of the cell, which is pretty astounding when you think about it. All right, so as we get into cell structure, we do kind of get bogged down in a review of the cell organelles. I'm not going to take class time to do that. The book covers them adequately. Most of you are probably already familiar with them. So what I'm going to tell you is that the list you see, which is reflected in your online materials, like the practice quiz on cell organelles, is going to tell you which ones you need to know. So for all of them, you need to know their name, you need to know their function, but you also need to be able to recognize them from images, okay? And there are a variety of images. This one I snagged off the internet, and you can see two different views here. So one of the views you see here is sort of a nice three-dimensional rendering, and the other one that you see is just a little line diagram, okay? So there are lots of ways that these things can be imaged. This particular one is actually an electron micrograph that I took myself when I took a course in electron microscopy. And it says pre-class assignment check, but that's only for the long term when we actually have time to do that. So what you see here is the nucleus in an electron micrograph. And you need to learn to recognize certain aspects of that. First of all, what is the nuclear envelope? What's the nuclear envelope? It's the boundary. Remember we talked about organization and how things are organized with boundaries. So the membrane that surrounds the nucleus. The nucleolus, those of you that have had intro A&P, I know you know what a nucleolus is, right? Yeah, it's that little dark spot there. The nuclear pores are openings, which you can see kind of as little holes here and there. And then the chromatin is all the stuff that fills the nucleus. The chromatin is the DNA. Okay, so that's the level at which I expect you to review these organelles. Make sure you know them. Make sure you know the parts. And look at them from different images. You have some images in your textbook. I just Googled some. You can Google labeled cell images and make sure you're looking at animal cells and not plant cells or bacteria cells. But you need to finish this review, okay? And again, I will post this PowerPoint outline for you so that you'll have a list of what structures you need to know. We talked yesterday about the phospholipid bilayer, the structure of the membranes of a cell. Y'all remember we did that? It was just like not even 24 hours ago. But we didn't talk about everything in the plasma membrane. We only talked about the phospholipid bilayer. So now we're going to tackle the rest of those. And we are going to spend a little bit of time on making sure we're familiar with those because we have an application for that. So in addition to having the phospholipid bilayer, there are also lots of proteins. Some of the proteins are transmembrane. What do you think it means to be transmembrane? Yeah, no doubt. But in terms of cell structure, what do you think that probably means? You realize I'm recording this, right? Okay.
I'm sorry.
Just saying. Transmembrane means it goes all the way through, okay? So some of these proteins, if they go all the way through, they are also embedded, and we call those integral proteins. So if it's an integral protein, it's at least partly embedded in the phospholipid bilayer. And if it in fact goes all the way through, then it's a transmembrane. Okay so far? Yeah. All right, good. Glyco is a word root that means sweet. Gluco or glyco refers to sugars. They're sweet. So we use the term glyco to refer to carbohydrate chains. And here you see carbohydrate chains attached to protein. So that makes it a glycoprotein. We also will see some glycolipids. What do you suppose that means if it's a glycolipid? It means that you can have little carbohydrate side chains attached to what would otherwise be a phospholipid. It's a glycolipid instead of a phospholipid. Okay? So these carbohydrate side chains are always going to be on the outer face of the membrane. They never point into the cytoplasm. They always point out to the outside, and we'll talk about their functions. Now, these terms, hydrophilic and hydrophobic, you should be okay with, right? Okay, so we already looked yesterday at the fact that our little fatty acids are hiding away from water in the middle of the membrane, and the little yellow circles represent the phosphate hydrophilic heads of the phospholipids. Everybody pretty clear on those? All right, so we'll also find that there's going to be some cholesterol in that membrane. Where would you expect the cholesterol to be? In between the heads? Is that what I heard you say? Let me see if I can change color. So if cholesterol is about this size, where am I going to stick it? Am I going to stick it up here between the heads? Is it hydrophilic or hydrophobic cholesterol? Y'all feel free to look back through your notes yesterday if you took notes. It's classified as a lipid, which means hydrophobic. Okay, so which part of the membrane is hydrophobic? The middle part. So your cholesterol sits like right in here, not out among the phosphate heads. Yeah, I heard what you said. You said between them, but there's between like left and right and between up and down. So that's why we're exploring this more fully. Okay, one little part of the cholesterol is polar. It has an OH group stuck to the end. I'm sure y'all remember I showed you that yesterday. So that little part would kind of hold the cholesterol closer to the phosphate head. Not by much, but by a little bit. So I can put some cholesterols here. They're found on both sides. And what they do for you is they get in between those fatty acid tails, and they kind of do two things. They space them a little further apart, but they also kind of hold them. And that keeps the membrane from packing too tight, but it also keeps it from flying apart. So it keeps it just right in the Goldilocks zone. Not too far apart, not too close together, not too solid, not too crazy far apart, just right. And it keeps the membrane fluid, and that makes the membrane more stable. Okay, so where is the cholesterol found? It's in that hydrophobic region. What does it do? Helps stabilize the membrane. Okay? By the way, just out of curiosity, I can point out that cholesterol is found only in animal cells, not in plant cells. So when you see plant products advertised as being cholesterol-free, yeah, cholesterol isn't found in plants. So, just saying. Okay. All right, so we have, if we look at the chemical composition of the membrane, we have the lipid components. We talked about the phospholipids. We talked a little bit about the glycolipids, and we've finished pretty much talking about cholesterol. So one more thing about cholesterol, just to nail it down. Is cholesterol good for you, bad for you, or what?
It's hype.
It's all hype, right? It's not all hype, but there is some hype to it. So is high blood cholesterol a health risk? Yeah, probably, at least for some people. Is dietary cholesterol correlated with high blood cholesterol? The more you eat, the higher your blood cholesterol is? Actually, no, it's not. So they used to, back, probably decades ago, they said you shouldn't eat that many eggs. Why not? You shouldn't eat eggs every day because eggs are full of cholesterol. Okay, but they finally figured out that dietary cholesterol, what cholesterol you eat, is not directly correlated with your blood cholesterol. So they've loosened up just recently on eating eggs. If you have high blood cholesterol, they're no longer recommending that you lower your consumption of cholesterol. They're suggesting there are better ways to lower your cholesterol, the number one way of which is not eat so much, and the number two way is get more exercise. Okay, our proteins, we have our integral proteins, some of which are transmembrane. The other kind of protein we have is called peripheral. Where would you look for something that is called a peripheral protein? On the periphery, maybe? So if this is a transmembrane integral protein that I'm drawing, where would I put a peripheral protein? Yeah, so you just kind of park it on the heads. Generally on the outside. I check every few years to see if anybody's ever found something they'll call a peripheral protein on the inside. No. Generally on the inside, not the outside. Peripherals on the inside. The ones on the outside generally are not attached. They're not truly attached. So they're not considered peripheral proteins, inside only. And generally speaking they're going to be like, they're going to have some enzymatic activity, or in some way they're going to activate something inside of a cell. And then last, our carbohydrate group. What was our word root for carbohydrates that let us know we were looking at that? We had glyco. So those glyco groups, those carb groups, can be attached to a protein or they can be attached to a lipid. So we have glycoproteins and glycolipids. Either way, those carbohydrate groups are in fact on the outside of the membrane. And there they're going to make a sort of a coating on the surface of the cell. You could think of it as sugar coating the cell. And it's called the glycocalyx. Glycocalyx is the coating of carbohydrate on the outside of a cell. All right, focusing in on our phospholipids. We just did this yesterday, so I'm going to kind of go fast through this, but if you have questions, let me know. Y'all remember that we had this part on the top that was called the phosphate head. And I told you yesterday there's a phosphate group, but there's also a nitrogen group. Okay, nitrogen groups are polar. Phosphate group is ionic. I've done something that's made my device unhappy. Okay, and then you also have the hydrophobic region, where the fatty acids hang out. Which one of those two fatty acids is unsaturated? The kinky one. So we always have one that's fully saturated and one with a kink in it, and that kink occurs where we have a double bond. Okay, so the way we tend to abbreviate our phospholipids is with the little circle indicating the phosphate head, then the zigzaggy lines indicating the fatty acid tails, and we know how to make a phospholipid bilayer, right? Any questions about that? Okay, and what about our cholesterol? Where does it sit? sits adjacent to the phosphate heads, but tucked in between the fatty acids. And what is it doing for you? Spacing out those fatty acids so that the membrane has the right fluidity or flexibility. All right, looking at our proteins, which one, where is the glycoprotein part in this picture? It's the green things up at the top. So there's a carbohydrate group, there's a carbohydrate group attached to a protein. You remember yesterday we talked about how proteins tend to coil? So here you see a coil, and then a fold, and another coil, and another fold, and another coil, and then a part down here. So that's typical of the shape of a protein. Is that an integral protein or a peripheral protein? Definitely integral, right? What about this red thing right here? That's another protein, and that's a peripheral protein. Very often those peripheral proteins that are along the base of the membrane like that are involved in either structural support or maintaining the shape of the molecule. Oh, I hit end instead of next. Okay, there are lots of kinds of proteins, and they have different roles to play within the membrane. So we've got, looks like 1, 2, 3, 4, 5, 6 of them here. Let's see what we can do with those. Let me get back to where I was. It's not letting me do my thing anymore. Okay, I may just have to do it from here. There we go. There we go. Okay. All right, so what you see across the top of the slide is some, it's done it again, is some, I may just have to give up on this completely in just a second, and I'm sorry if that happens. There we go. You've got some transmembrane proteins here, and they have a variety of functions. So you see the labels across the bottom. So you have receptor, enzyme, channel, gate, cell identity marker, motor molecule, and cell adhesion molecule. Does everybody see that list? Okay. So you need to be familiar with these so that you can explain what's going to happen with a cell in terms of which protein's going to do what. So what do you think a receptor is going to do? It receives. So very often we have a chemical messenger. The proper term for this is ligand. Some people call it ligand. Ligand. A ligand is a little chemical that can either bind or release. So it's kind of like, in my opinion, it's kind of like putting a finger on a doorbell. So it's like ringing the doorbell. So when the chemical binds to the receptor, then something in that cell is going to react. That receptor causes some change. And very often the change is to activate an enzyme. But sometimes the receptor will cause a gate to open. So I want to talk a little bit about channels and gates, and we're going to talk some more about those in more detail later. The term channel versus gate is unfortunate in my opinion, because a gate is an opening to a channel. So instead, I'd like you to refer to the channels as either having gates or not. They're either gated channels or they're ungated. Okay? And we're going to come back and explore that some more. What do you think a cell identity marker does? And what about that thing that's attached to it? What is that? That's that carbohydrate. So one of the jobs of the carbohydrate is to provide a molecular identity for the cell. So you've heard of blood types, right? You know if you're blood type A, B, O, or whatever. Those molecular markers that determine your blood type are carbohydrates on the surface of your cells. They happen to be on the surface of your red blood cells. They also happen to be on the surface of a lot of your other cells. So you could actually scrape some cells off the inside of your cheek and use those to determine your blood type because you have the same markers on those cells, okay? So they're not the only things that mark cells, but certainly a lot of the things that mark cells are carbohydrates. And then finally we have CAMs. A CAM is a cell adhesion molecule. And here you can see the CAM of another cell. So what are we looking at there? We're looking at an extreme close-up of where two cells are connected to each other. So anytime two cells are physically connected to each other, they're using some kind of integral protein to lock themselves together, a CAM. Okay? So does everybody have this list? Receptor, enzyme, channel, whether it's a gated channel or not, cell identity marker. We didn't talk about motor molecules. I'm not real worried about them. And the cell adhesion molecules. Got it? All right, let's see if we can move on. Okay, so let's see what we have here. I have a very large blue protein with a white arrow on it. Do you see that? So what is that? This arrow right here. Let me change to yellow. This arrow right here, pointing to this big protein. Can you give me a specific category for that protein? What would that one be? A what? I'm not sure you can tell whether it's a gate or a channel, but certainly you could tell me by its position whether it's integral or peripheral, right? Okay, so which one is it? Integral, definitely integral. Is it transmembrane?
Yes.
Yeah, it sticks out both sides, right? What about the other big blue protein on the other side? This big blue protein here? Doesn't it do the same thing? So if it projects out one side and out the other side, it's transmembrane, okay? If it's transmembrane, by definition, it's integral, okay? Because integral means at least partly stuck in the membrane, okay? So what about the orange one here? That's another protein, it's just a different protein. Is it transmembrane? No, it doesn't stick out both sides. Is it integral? Is it embedded at least part of the way in that phospholipid bilayer? Yeah. So it's integral but not transmembrane. Is everybody okay with that? All right, and then I'm also pointing to these little green things, which I didn't really talk about. Those are cofactors. A cofactor is a non-protein substance that is involved in a protein being functional. Okay, and chances are we're not going to have time to get to that. All right, so here is our first applications. For each table, what I want is one sheet of paper. We'll see if it takes more than one, but write small. At your table, if you're not sitting with a group, move to join a group. At each table, just start a piece of paper, first and last names of everybody at your table. And I want you to sketch just the phospholipid, cholesterol, and a protein channel. You guys can decide for yourselves if you can stay on the side of each other or what. Remember, if you need paper, I have it up here. And I'm giving you a 3-minute limit. All right, so part of that is just to give you a mental break from just listening to me. Part of it is so that I can assess whether you are catching on. So what did your pictures look like? Can't wait to see them.
Pretty bad.
Being able to draw is a skill worth developing. I am a crappy artist. Y'all can tell by how poorly I write. I'm really good at stick figures. That's about it. I have a brother who's a professional artist. I got none of those genes, apparently. None of them. And I have some very dear friends. Miss Gregory is the artist for our lab manual. So, so I'm no good at that. But I can tell by looking at your pictures whether you get it or not. So drawing's important. Drawing is an important form of communication, and it helps you to be able to draw well. So when you look at your drawings, if they don't make sense to you, they're not going to make sense to anybody else. And yes, labeling is crucial, making sure that you can look back later and see what those labels mean. All right, so we're going to move on to cell membrane functions now that we've kind of nailed down the cell membrane structure, sort of, kind of. And we're going to come back to one of our themes, complementarity of structure and function. And we're going to explore that to some degree. We're going to talk about 4 different membrane functions. We're going to talk about one of them more than the others, membrane transport. And that's what you do. That's today's lab, isn't it? Isn't today the cell lab? Did y'all get to cut cells this morning?
Yep.
Did they do it as a competition?
Yep.
Did anybody in this class win? Anybody? Anybody? Second place? Hey. All right, so you can talk to the afternoon guys about strategies if you need to, but we'll leave that for lab. So here are 4 big ideas. Membranes keep the cell organized. They compartmentalize the cell. They allow the cell to have different areas with even the pH can be different in one compartment of a cell than another. So the organization of a cell is based on the compartmentalization due to the membranes. Selective permeability has to do with allowing stuff to come in or go out of a cell. Cells control what comes in and goes out. And we'll look at those processes in lecture today, even though I know you covered them in lab. Cell adhesion, we looked at those cell adhesion molecules, the fact that cells have to be able to attach to each other. Doesn't that make sense? Would you be able to stay together as an individual if your cells were not stuck together? So that one's pretty obvious. And then cell signaling has to do with how cells communicate with other cells. We'll be talking a little bit about that, and that one definitely we'll come back to later in the semester. So we've already looked at our structure, right? So just a quick review. What would this be? This big red thing right here. That's some kind of a channel, right? What about this thing right here? That's just a peripheral protein of some kind. Who knows what? What about this right here? That's your carbohydrate group that makes it a glycoprotein. What about this one? Is it integral or peripheral? The green one. It's integral, but it's not transmembrane. Very good. Okay, so we have some kind of an idea of what is going on. Now, we're going to look at the pathways through the membrane, and I use this term very particularly because if I don't, students tend to muddle up the processes by which things move with the direction by which they move. So just a big idea is the difference between a pathway and a process. Okay, so if I asked you how to get from here to the library, what are your pathways? What are your choices for pathways? You just have to get from here to the library. What are your pathways? Take sidewalk.
Straight line.
Yeah, there's a nice straight line. You would have to jump over a fence if you wanted to go straight line, but that's okay. So you could go cut through the grass. You could, I suppose, climb a tree. That's what the squirrels do. Does everybody understand the idea of pathway, though? If I asked you what process you would use to get there, what are your choices for processes? You could walk. You could ride a bike. We have people with those little motorized scooters around sometimes. So that's the difference between pathway and process. Does everybody understand that? Okay, so the pathways through the membrane include things that can go right through the phospholipid bilayer or things that need a transmembrane protein to get through. Honestly, that's the only 2 choices you have. There are different kinds of transmembrane proteins, but you're either going to go through a protein or you're going to go through the lipid bilayer. That's your pathways. Are you with me on that? Okay, so then we need to understand a little bit about the chemistry of what kinds of things can go through the phospholipid bilayer, because if they can't, then we would know to predict that if they're going to go through the cell, they better have a channel of some kind. Make sense? So if it's going to go through the lipid part, then does it make sense it has to be lipid soluble? Lipid soluble means it mixes with lipids, which means it's nonpolar. Okay? Do we know anything that's small and lipid soluble? Small enough, fats, big molecules, too big, but something that's small and lipid soluble includes, believe it or not, cholesterol. Even though we know cholesterol tends to be embedded in that membrane, it can go through the membrane. And a couple of things on your list of small and lipid soluble: oxygen gas and carbon dioxide gas. Okay, so if I asked you, what pathway does oxygen use to get in or out of a cell, what's your answer? What pathway does it use? Goes right through the phospholipid bilayer. That's the answer to the pathway. Make sense? Okay. If it's small and hydrophilic, not lipid soluble, ionic, or a small organic acid, then it can't just zip through the phospholipid bilayer. It can't do that because lipids repel things that are hydrophilic. So for those guys, we need a channel. And the thing about our channels is that they are tailored to allow the passage of some substances, which means other things are not going to go through. So we have channels that are specific for specific things. And we name them for the things they're specific for. So guess what goes through a sodium channel versus a calcium channel versus a potassium channel? If I tell you there's a channel that's a sodium-potassium channel, what do you know? It lets both sodium and potassium through. So this is not hard to figure out. We name them in a way that makes sense. So there are, though, cases where more than one thing can go through a channel, and we take it for granted. There are also cases where one kind of channel lets water through and another kind doesn't. And I don't expect you to know that unless I tell you. Okay? So I'm not expecting you to memorize all of that. But there is a term for water channels, and it's not a hard term to figure out. They're called aquaporins. And aquaporin is a water channel. Aquaporin, all one word. I just ran out of room. Why does water need channels?
To supply the muscle.
Well, just to get in and out of a cell, not looking, you're thinking big. I'm just thinking in terms of the logistics of the cell. Why can't, water's tiny, isn't it? Little teeny tiny water molecules? Isn't it small enough to slip right through those phospholipids? Yeah, exactly. Those membranes are hydrophobic, right? So they repel water. So even if a few water molecules could sneak through, not enough to be physiologically useful. So we need to have those water channels. And there are cells, for example, in your kidneys that don't have them. Does that make sense to you, that your kidneys can control water by some cells having water channels and other cells not having water channels? And actually some are regulated by hormones. The presence or absence of water channels is hormonally regulated in the kidneys. Okay? So water in general does not freely flow in or out through the lipids. Just like everything else that's hydrophilic, it needs a channel. Are you with me on that? Okay. So that's our two basic choices. As we'll see, there are some other choices. So if you want to write this down, that's fine. Remember, I'm going to give you this list later. But we have these pathways and these processes. All we've looked at so far are the pathways, although in lab I know you already looked at the processes, right? So what are your choices for pathways? Something can go through the lipid, or it goes through a protein. And for the proteins, remember these have to be transmembrane proteins. Some of them we call channels, others we call carriers. And the distinction between a channel and a carrier is not that clear cut, so I'm going to oversimplify it for test purposes. I'm going to make sure you know what to call a channel and what to call a carrier. Okay? We're just not going to worry about it other than that. And then our processes include diffusion and active transport. And we have two kinds of diffusion. We have simple and we have facilitated diffusion. So if I asked you what pathway does oxygen use to get in or out of a cell, what was your answer for pathway? Goes through the lipid bilayer. But if I ask you what process oxygen uses, does anybody know the answer to that? It's simple diffusion. Okay, so asking you how does oxygen get through a membrane is a vague question, right? Do you mean pathway or do you mean process? So y'all got to learn to think in those two different venues so that you're always aware of what the difference is. Let's see if I can get this to work. I'm wondering if I can make it big enough to be useful. Okay, I should have vetted this before I started. We're so thoughtful. All right, I'm going to have to post that link online for you later. Okay, sorry about that. That's not going to work on my Mac. All right, so here you see the beginnings of a list of things that you need to remember. The full list is available to you in the chapter summary, and it's also in the quiz on Canvas. But you can go ahead and write it down if you want, so you'll have it for reference. I already gave you the small lipid-soluble guys. That's oxygen and carbon dioxide. There are others. Small ions and water need to use channels. We may add to that ion list, but that's probably pretty close to being complete. And we need to talk about this third category, which is carriers. Carriers are simply a second kind of transmembrane protein that are very specific. And we have carriers that will carry ions for active transport. And we also have carriers that will pick up our small organic molecules, which includes, but is not limited to, glucose, amino acids, and lactic acid. And then our fourth category up there is a vesicle. A vesicle is a little membrane-bound sac. It looks like a little bubble inside of a cell. And you use vesicular transport for things that are too big to go through a channel. So there are a lot of cells whose job it is to produce and export proteins. Proteins are big. How would I get proteins out of a cell? I have to use vesicles to do that. Okay? There are other proteins that need to get into cells. So again, we use vesicles to bring them in. All right, so does everybody see that we have on the beginning, we have our pathways, right? And then for each pathway, we have a list of things that use that specific pathway. Does that look like a set of test questions to you? It sure should. I do expect you to remember these. Okay, questions about that? All right, so we're ready to talk about our processes. And again, you're going through these to some degree in lab, so I'm going to kind of go fairly quickly through them. Diffusion is defined as the movement of solute particles along their concentration gradient. That is from high concentration to low concentration. Simple diffusion is diffusion through the membrane, either through the lipid layer or through the channels. That's simple diffusion. So if it goes through the lipid bilayer, or it's got a channel that's sitting there ready to go and it's just diffusing along its concentration gradient, that's simple diffusion. Okay, so anything that goes through the lipid layer or uses a channel is going to enter or leave a cell by the process we call simple diffusion. Got it? Okay. If it needs a carrier, and which things needed a carrier? Ions only for active transport, but our small organics like glucose. need carriers. If it needs a carrier, the processes that we use are lumped together as carrier-mediated transport. Carrier-mediated means nothing more than it needs carrier. So what kind of carrier-mediated transport depends on which direction it's going. And the direction is based on the concentration gradient. If it's going along its concentration gradient the way it would diffuse anyway, but it needs a carrier, then we call it facilitated diffusion. Okay? If it's going against the concentration gradient, you're actually pumping something into a cell, then that's active transport. So active transport is the opposite of facilitated diffusion. Is everybody pretty clear on that? So we have two carrier-mediated processes. They go in opposite directions of each other. One goes with the concentration gradient, the other one goes against the concentration gradient. Pretty clear? Any questions? We're going to apply that, so hold on to that thought. Our last process is called vesicular transport. That means it's using vesicles. And it's either coming in or it's going out. If it's coming in, we call it endocytosis. If it's going out, we call it exocytosis. Okay? And if we have time, we'll look at some examples of that. I have to say, of all the kinds, that's the one that we use the least. It just doesn't seem to be an opportunity in this course much to go, see, here's why you needed to know vesicular transport. It just doesn't seem to happen. All right, so I'm not sure if my animations are going to work. We're going to see. Cannot open the specified file, so that's that. You're going to study this in lab, right? Okay, so we'll hold off on that and we'll see how that works. This one, likewise, I probably didn't save my animations. These animations are also embedded in the chapter summary for chapter 4. Okay, so look for those animations when you go through that chapter summary. Okay, so just as a general statement, if it is lipid soluble, what do we know about it? If it's a lipid soluble substance, what's it going to do? Just zips right through the lipid bilayer. If it's water soluble, what does it need? A channel. Okay? And again, depending on the diffusion gradient, that's going to determine the direction, right? All right, that's as good as it's going to get today. All right, osmosis. How is osmosis different from any other process? Osmosis specifically refers to diffusion in the body of water. It in more general terms, is always the diffusion of whatever the liquid is that stuff is dissolved in. In our case, that's always water. So osmosis for us is always the diffusion of water through a selectively permeable membrane, meaning some things can diffuse through and some things can't. Okay? So in the case of osmosis, you're always comparing two sides of a membrane. Here's a cell. And the water can easily go, but I've got some solute that cannot get across the membrane. Is that all right with everybody? Generally speaking, cells have more stuff in them than the extracellular fluid does. So the easiest way to envision osmosis is simply to look at it in terms of what's the percentage of this fluid that is composed of water. Okay, so the more stuff I have, the higher the concentration of stuff, solute, in the cell. So if the solute concentration in that cell is, say, 1%, what's the concentration of water? 99%. If it's 1% solute, it's 99% water. So what if out on the outside, it's only 0.5%? What's the percentage of water there? 99.5. Doesn't sound like a big difference, but it's enough to make a difference. Which way will water diffuse? If there's more water, if it's 99.5 on the outside and 99 on the inside, which has more water? The outside compared to the inside. So which way is water going to diffuse? Does everybody get this? If you don't get it, just look at me funny. If it's, let's do a simple case. If I have a selectively permeable membrane and this side is 10% and the other side is 20% solute sugar solution, 10% sugar on one side, 20% sugar on the other, which way will the water move?
Right.
Left to right or right to left?
Right.
Left to right? Which one is more water? The right side is more water, right? So it's 90% water, whereas the other side is 80%. And water, it's not accurate, but it's good enough for us. Water can be thought of as diffusing along its concentration gradient. So if I have more water on the right than I have on the left, which way is the water going to go? From high to low. Water, like solute, will move from high to low. And the other way to think about that is that water is always going to be attracted to the solute. Wherever there's more solute, that's where the water goes. Water follows solute. And that's a handy way to think of it because the cell is able to move the solutes around. So wherever you park the solute, what does the water want to do? Follow it, wherever it goes. Okay? So back to this case. If this is 0.5% in the extracellular fluid and 1% on the intracellular fluid, which one has more solute? The inside or the outside? Inside has more. Water goes wherever there's more, so water will tend to flow in. Okay? Now, what did you do in lab this morning to nail that concept down? Who had lab this morning? You had eggs in lab, right? And some of them shriveled up and some of them swelled. And you need to keep track of the meaning behind that as well as the terminology associated with it. Okay? So that's just a brief introduction to osmosis. Make sure as you study for lab and as you use the online notes that you really feel comfortable about that. People die when mistakes are made with not controlling the tonicity, the osmotic concentration of fluids that are being delivered intravenously. Okay? So as soon as people can die from something, that puts it on my list of important things to remember. Is that a reasonable way to measure importance? Okay. So y'all feel like, you need to feel like you've got that nailed down. All right. On to carrier mediated. There's some terms here. I keep forgetting to hit my pen. If one substance is being transported, that's called uniport. Uniport. So, for example, your thyroid gland is the only place in the body I know of that sucks up iodine. So it has a little iodine transporter, and it takes just that one substance in one direction. That's a uniport. If it takes two substances together, that's symport. If it pumps two things in opposite directions, that's antiport. And if it can pump two different substances, but either one or the other, that's called competitive. You will probably not find competitive in your books. But I'm going to give you a case where competitive transport actually has clinical applications. The competitive transporter that I'm familiar with will carry either calcium or magnesium. Now, looking up at your periodic table again, do you see calcium and magnesium are like one right on top of the other? They're both in column 2. Magnesium is in row 3 and calcium in row 4. So they're similar in size and they're identical in ionic charge, which means that it's very easy to imagine a protein of a certain size and shape that could pick up either calcium or magnesium and transport either one. Their properties in body fluids are very similar to each other. There are enzymes that can recognize just magnesium, and there are certainly pumps that will pick up only calcium. But in your duodenum, the first part of your small intestine, you have a competitive transporter, a calcium-magnesium competitive transporter. And here's why that matters. If it's competing for calcium versus magnesium, if you overload your diet with too much calcium, then calcium can outcompete the magnesium for this transport process. And people do that. So a lot of people take calcium supplements. Why do people take calcium supplements? Why do most of us take that? Bone health. We talked about that just yesterday or the day before. Can't remember when. First day. Calcium for bones, right? Makes your bones stronger. So it turns out that in the world of competitive basketball, that there were some, and these were pro basketball players, that were taking excess calcium to make sure they didn't get bone fractures. What kind of bone fracture is a basketball player likely to get? Anybody know? It's usually their heel fracture. Why? Because after they're going up, they're landing on those heels pretty hard. And so it's a known occupational risk for basketball players. Heel fractures, they'll do anything to avoid them because that could be a career-ending injury. So they're taking these calcium supplements, right? And we all know that in the general, people know that if one's good for you, two must be twice as good, right? So, yeah, y'all know better than that. But these guys didn't, so they were overloading on calcium supplements. And how do we know that this was a problem? They had heel fractures. So they're like, how did I get a heel fracture? I was loading up on calcium. Well, guess what else bones need for their health? Magnesium. And so they were overloading themselves with so much calcium that they literally had a deficiency in magnesium because of this competitive carrier. So it matters, and you need to know about it, okay? All right, we'll talk about the others as we get into some applications. So, and then, of course, with the direction of movement, we know that if it's with the concentration gradient, it's facilitated diffusion. If it's going against the gradient, it's active transport. Questions at this point? All right, vesicular transport. Very quickly, just some terms. If we are bringing it in, it's called endocytosis, taking stuff in. If we take it in fluid, that's called pinocytosis. If we take it in large clumps, that's called phagocytosis. So the nicknames for these, pinocytosis is sometimes called cell drinking, and phagocytosis is called cell eating. Cells can eat an entire other cell by phagocytosis. Your liver cells and spleen cells that are responsible for getting rid of your old red blood cells just gobble up the whole cell, phagocytosis. Okay? If you're just sort of sucking in some fluid, though, and there's some protein suspended in it, that's pinocytosis. And then our third one is a very specific process. Receptor-mediated endocytosis is used, for example, when antibodies bind to their antigen and activate your immune system. You don't want them reacting to just anything, right? So they're very specific about what they react to, and that makes it receptor-mediated. Now, does anybody have a clue what transcytosis might be? We know what transmembrane means, right? Goes all the way through. So in transcytosis, you're taking something in on one side of the cell, letting it cross through and get out the other side. So just passing through a cell, coming in one side and going out the other side, and we see that in our capillaries. Questions about that? All right, and then exocytosis is secretion from cells. All right, so that is kind of our review. So let me restart this for you. This is going to start my clock. For each of the, use the same sheet of paper, and for that list of things, see if you can come up with the list on the left. And while y'all are doing that, I'm going to go see what he did.
Oh, it's for oxygen. Pathway for oxygen. So, that's the sodium ion.
How are we doing on our list? Bad? I just gave you all the answers. Who's messing with me? Okay, so does that look like information that might show up on the test? Yes. Yeah, pretty much. Okay. So definitely feel like there's hot air coming out of it. It does, doesn't it? All right, so were any of them hard? Did you get stuck on any of them? We got oxygen, right? That one's easy. You got one of them? Good, good. Hey, you have till Monday to get the rest, right?
There you go.
There you go. So glucose, what about glucose? It's a small organic. Needs a carrier for small organics. Sodium ion? Does it have to be a channel? Can't it also use a carrier? Yes, it can. Okay, water? Aquaporin, which is what category? It's a channel. Okay, what about a protein hormone? It needs a vesicle. Protein hormones have to be picked up by vesicles if they get in or out of a cell. Remember, they could be leaving the cell as well as entering the cell. Okay, and I told you up there, I gave you a big hint. It's a large fibrous protein. What about collagen? As a large fibrous protein, it does not go in or out. When you make collagen in your tissues, which you'll look at in lab tomorrow, you'll see how big they are. You actually secrete it as little collagen proteins, and then they get assembled into large fibers when they're already out in the extracellular fluid. And the reason I bring this up specifically is that people use collagen creams to treat wrinkles. How well is that going to work for you? If you put collagen on the outside of your skin, does it get absorbed? Sorry, no. It's not going to work. Okay? All right, any questions about that? Pretty good with all those? All right, this is actually covered in the chapter summary. I want to go through it one time with you in class and then send you, because I know my animations aren't going to work, send you to the online version to see it at work. Okay? This is a confusing story. So I want to take a little bit of time to go through it slowly with you, and I think I'm actually going to try to draw some pictures if I can get my drawings to work. Yeah, it's not. Again. And I like this story because it pulls together diffusion with osmosis, with active transport, and it also has a clinical application. So what does it mean? Those who've been to lab this morning know this term hypotonic. What does that mean? Does it have more or less? It has more water, less solute than blood. Okay, a hypotonic solution is more water lost than it is salt or other solutes. Okay, so we're going to look at how the body manages to do that, and then we're going to see what happens when things go wrong. Sweat is initially formed as an isotonic solution. What does that mean? Equal in concentration to blood. And then what we do is we sneak the salt back before the fluid is secreted. This avoids wasting salt. So you know that in certain extreme circumstances, people can go into heat shock. They get dehydrated. They've lost so much of their body fluids from sweating that they have put themselves in a really bad situation. And that's basically what we're going to explain how the body works to prevent that from happening. All right, so looking at a sweat gland. When you look at a sweat gland, what you see is that the glands sit down here at the very base of the skin, and then they have a duct that empties out, usually in a hair follicle, although not always. It's going to empty out onto the surface some way. Okay? That's one kind of sweat gland. Here's another. Again, it's just sort of a coiled up tube with a duct that opens out on the surface of the skin. All right, so that's what the gland looks like. What we're going to find is that there are two distinct parts to this. The deeper part of the gland itself has a role to play, and then the duct has a separate role to play. So we have the secretory portion that actually produces the sweat, and then higher up we have the duct to transport it onto the surface, but we're going to find that the duct also is an active player. All right, so here is an extreme close-up of a single cell. And in this cell, what you see is an aquaporin, a chloride channel, and a sodium pump. Okay, so what do you know at this point? Cells on the left, the duct or the lumen, the hollow interior of the gland is on the right. The arrows tell you which way things are moving. What am I doing first? I'm going to pump sodium. I'm going to use active transport to deliberately push sodium out of the cell. I'm going to use ATP and active transport to do that. Okay? What's going to happen next? If sodium is out in the extracellular fluid, it has a positive charge. And that electrical charge will do what? It's going to attract the chloride ions because they're negatively charged. So chloride will freely and simply diffuse to follow the sodium. I don't have to pump the chloride. All I have to do is pump the sodium, and the chloride will follow. Does everybody see how that works? It's an electrical gradient. It's not a concentration gradient, but it's an electrical gradient. Are you with me so far? Okay, so what about the water then? Why is it going? It's following the solute. Remember in osmosis, water follows solute. So if I pump the solute, I can push the water wherever I want it to go. So basically what I'm doing in these sweat gland cells is I'm pumping sodium, chlorine follows the electrical gradient, the chloride ion, and then water follows by osmosis. Is everybody pretty clear on that process? So your sweat gland cells are sitting there doing this. At a base rate, they do it pretty much all the time. We call that insensible perspiration. You can't really feel that you're sweating. You're doing it. And as that water then gets pushed out to the top of the surface of the skin, when the water molecules evaporate off, they carry heat with it, and that's how you thermoregulate. All right, so that's the basic story. Now, what we find in the duct is something a little trickier. What do we see going on here? Remember, we're in the duct now, not in the gland. So what we have coming up, we have sodium that we pumped, we have chloride that we allowed to diffuse, and we have water. And sodium, chloride, and water are all moving up in this fluid. But what happens is that as it reaches the duct cells, the sodium gets snagged, some of it, not all of it, some of it gets snagged and pumped back in. And then, of course, that creates an electrical gradient that makes the chloride also diffuse in. Okay? What's missing in this picture? There's no water channel. There's no aquaporin. So as a result of this, as this fluid moves up and out, more of the water and less of the salt is still contained in that fluid. Okay? So I reclaim my salt. I don't waste my salt. So what was the point of pumping that salt? Why did I bother to do that if I'm just going to pull it back in again? I'm pushing osmosis in the way I want the water to go. So I'm doing this strictly to control the osmosis. Can I save all the salt? No, I cannot. Some of it will inevitably be lost. But I can save some of the salt. Does everybody see how sweat occurs? Questions about that? So which part of this is active transport? Sodium gets pumped. First out, then back in. What about the chloride? That's simple diffusion, right? It's going through a channel. And then the water, osmosis. Okay, so does everybody see how we combine those three processes? And this probably is not going to work, darn it. But if it doesn't, it will definitely work. Yeah, can. It'll work when you go to the web page. All right, so here is our clinical application. In cystic fibrosis, there's a genetic mutation to some of the chloride channels. And it turns out that these chloride channels are found in different body systems. I'm going to give you about 3 other locations. One is in the respiratory tract that we find these defective chloride channels. And this is why kids with cystic fibrosis have percussive therapy to get mucus out of their lungs and tend to have lung infections. Okay, so these defective chloride channels are in the respiratory tract. They're also in the pancreas. And in the pancreas, the defective chloride channels prevent the pancreatic juices from flowing properly. And those are involved in digestion. So kids with cystic fibrosis have extreme problems just simply digesting food. They have to swallow extra enzymes, and you have to beef up their diets to get enough calories. So kids with CF tend to be small, underweight. We call it failure to thrive. They're just not doing very well. They tend to have respiratory infections, and they just don't gain weight. And all of that is due to this defective chloride channel. A third location is in the liver. And the liver, like the pancreas, relies on these chloride channels to help move the water along so that the liver secretions are appropriate. In the sweat glands, it's a little bit different. Because in the sweat glands, we have 2 different directions of our chloride channels, right? So let's look back at our pictures just to make sure. We see that. So in the gland, which way is the chloride channel going? It's letting the chloride out. But in the duct, it's bringing the chloride in. And these are literally different kinds of channels. Don't ask me to explain it. Okay, so we have in cystic fibrosis, it's the ducts where the channels tend not to work. So looking again at that picture, if the duct is not working, or if the chloride channel in the duct is not working, then what else is not going to work? if I need chloride to follow the sodium to maintain electrical balance, then I can sit there and try to pump sodium all day, but if the chloride won't follow it, I can only pump a small amount of sodium. So in effect, if that chloride channel is not working, then neither my sodium nor my chloride is going to be pumped. Okay? So that being said, what does that mean about the sodium and the chloride? It's in the duct. We're trying to reclaim it. So if we can't, then it goes out with the water. So tell me about the sweat in someone with cystic fibrosis. It's salty. It's extra salty. In fact, this is the first test we use to screen for cystic fibrosis. It's called the sweat test. And in fact, very often parents will know before they take the kid in for a checkup. Say, boy, I kissed their little sweaty heads and they're just salty as could be. So it's that much more salt. And so that's usually the first sign. That plus the fact that they tend to be underweight and they don't gain weight regularly and they tend to have respiratory problems. So the respiratory secretion, the pancreatic enzyme, and the liver secretion is all the opposite of what's happening in the sweat gland. In those cases, it's the chloride channel that lets the chloride out, which lets osmosis occur, that causes the problem. Okay? Does everybody see how important it is to understand how active transport and facilitated diffusion and simple diffusion and osmosis all work together so that you can appreciate what's happening when things go wrong? Tomorrow, it'll probably be tomorrow before we get to it, but we'll talk about the genetic defect associated with the chloride channel so that you can have a deeper appreciation of the problems in kids with cystic fibrosis. Okay, so that is the most I can spend on membrane transport functions. I'm going to be sending you home, not yet, but I'm going to be sending you home to nail down those concepts. Use my chapter summary that I have posted. There, the animations will work. And make sure that you have a good, strong sense of what isotonic, hypertonic, hypotonic, that you're not getting those terms backwards. Use your lab manual in addition to the lecture materials to make sure you're comfortable with all those terms and processes. And then you can ask me about them tomorrow. Now, we have 3 other groups of processes that we're going to look at. Communication and metabolism, I think we'll be able to get through today, which will let us focus on, we'll probably not get through metabolism. There's quite a bit there. So we'll get a start on that, and we'll do probably protein synthesis and reproduction tomorrow, and then we'll be done. Okay, so we're going to talk next about how cells communicate with each other. And one of the ways that cells communicate with other cells is by secreting chemicals. Do you remember the term I gave you, ligand? Do you remember what a ligand was? Anybody? It's what binds to the receptor. So the ligand is the little chemical that is your finger on the doorbell. The doorbell is the receptor, okay? So a ligand is a chemical secreted by one cell to activate another cell. In fact, chemicals actually self-stimulate. So they'll secrete chemicals that stimulate themselves. They'll secrete chemicals that stimulate nearby cells of the same kind or nearby cells of a different kind. They'll secrete chemicals that get in the bloodstream. We call those chemicals hormones. Okay? So cell-to-cell communication can be same cell, a nearby cell, a tissue nearby, or a totally far-off target. That's the important thing to remember about cell-to-cell. So we have some terms here. If it's the same cell, it's called autocrine. What does auto mean? Auto means actually self. Okay? So an autocrine secretion is one that stimulates yourself. Okay, crine, C-R-I-N-E, means to secrete, basically. If it's other cells nearby, it's called paracrine. Para means alongside or beside. Okay, so paracrine, nearby cells. If it's cells at a distance, it could be a neuron secreting a neurotransmitter, or it could be a hormone being secreted and carried in the blood. Okay, so you're either stimulating locally or you're stimulating systemically. Those are your two categories. Local would be close by. Systemic would be using either the nervous system or the endocrine system to achieve your stimulation. Okay? So how do cells know whether they should respond? If it's a hormone being secreted in the blood, how do cells know whether they ought to respond to that hormone or not? They have a receptor. Only the target cells for that particular ligand will have the receptor for that. So how do cells of the thyroid gland know to respond to the pituitary hormone called thyroid-stimulating hormone? They have a receptor for that specific hormone. And the rest of the body, those cells don't have that receptor. Is that pretty reasonably clear for now? Okay. One of my favorite chemicals, and I don't mind confessing to this, one of my favorite chemicals is caffeine. Pretty reliant on that stuff. So why does caffeine affect the body the way it does? What does caffeine do? What does it do for you? Stimulates your metabolism. It stimulates your brain, helps perk you up, make you awake. What happens if you're addicted to caffeine and you don't get your usual dose and you have to withdraw cold turkey? What happens then? You get a headache. Well, not everybody does, but a lot of people do. Apparently, a lot of people know that. So that tells you there's something going on. Caffeine is doing something. Caffeine is a ligand. Caffeine is actually mimicking chemical ligand secreted by your body cells. It's one that we are going to be very familiar with. It's called adenosine. So caffeine mimics adenosine. And I would write on this if it would let me, but right now it's not letting me. I don't know why. Adenosine. Adenosine is the A of ATP. And you know ATP is the energy currency of cells. So if you're metabolically very active, or if you've just been awake for a long time, your cells are using a lot of ATP. Now, we use it over and over and over and over again. But cells have a mechanism to let a little adenosine leak out. And the amount of adenosine that leaks out of cells depends on their metabolism. And so the longer you've been awake, the more adenosine leaks out of your brain cells. And when that adenosine level gets high enough, you are fatigued. And you rest. You may not sleep, although generally that's the way that works. Okay? So guess what caffeine does? It's either going to enhance it or block it. Which one? Adenosine makes you sleepy. What does caffeine do? It blocks it. Okay? Caffeine is an adenosine inhibitor. It blocks the receptor for adenosine and prevents the adenosine from binding. And so the adenosine is sitting there piling up in your brain, but caffeine's blocking the signal. And so it doesn't get through. Does everybody see how that works? Okay, well, there's a problem with that because cells respond to the fact that their signals aren't getting through. And they do what we call upregulation. So if you are a brain cell and you know that adenosine signal's not working, what are you going to do? Hey, man, my adenosine signal's not getting through. I better make more adenosine receptors. So cells respond to the blockage of caffeine by building more adenosine receptors. And so what happens then? 1 cup of coffee isn't enough, right? So then what do? You drink more. So you up the caffeine dose. What do your cells do in response? They make more adenosine receptors. So you build up a tolerance to caffeine. This is true of many other drugs. You build up a tolerance because cells that are wired to get a certain signal respond when that signal is dampened by upregulating, making more receptors. Does that make sense? Okay, so what happens when you hit that cold turkey moment? Your cells are so chock full of adenosine receptors that when the caffeine's withdrawn, they're overloaded. So your nerves are jangling and you can't sleep and you've just overdone it. Okay, the headache is a different phenomenon, and I'm going to explain it. We'll come back to it in AMP2. Brain cells are not the only cells with adenosine receptors. The capillaries in your muscles also have adenosine receptors, and the arterioles that supply those muscles, those cells have adenosine receptors. So if your metabolism has been going for a while, or you're physically more active, your adenosine level in the blood will rise, and caffeine blocks that. And your cells respond by building more adenosine receptors. And so here we are on our way to caffeine addiction pretty quickly. So when you get that cold turkey withdrawal, then all of your blood vessels are sitting there with all these extra adenosine receptors, which are now being stimulated because you withdrew from caffeine, right? So you tell me what the effect of caffeine on blood vessels will be. Will it dilate or constrict? Well, let's do the easy one. What's the effect of adenosine on blood vessels? Is it going to dilate them to let more blood in, or is it going to constrict them? Dilate them, because adenosine is produced when your metabolism is high, which means you need more blood. So adenosine dilates your blood vessels. Caffeine, because it blocks adenosine, causes constriction of the blood vessels. Is everybody with me on that? So when you quit cold turkey, what are all your blood vessels doing? Popping out, dilating. And the ones in your dura mater are so dilated that it pounds on the little pain nerve endings in your head. You do not have pain nervings in your brain, but you have them in the membranes around the brain, lots of them. So as those blood vessels dilate in the meninges, boom, 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 it's hitting those pain nerve endings. And every pulse of your heart is a shot of pain in your head. Okay? So what's the cure? Of course, what's the cure for that? Don't quit caffeine. That's my advice. But it is probably, I think I read once, I can't remember the number exactly, but it was like 50% of post-surgical headaches are probably caffeine withdrawal headaches. Why would that be? 50% of post-surgical headaches? What is it? No eating or drinking before surgery. So if you weren't planning ahead, if nobody explained it to you, and you just went, oh, I can't even have a cup of coffee this morning because I'm having a tooth extracted or a lung removed or whatever, then that's what we hear. I have such a whopping headache, and they blame the anesthesia or they blame the surgery, and if you would just give them a cup of coffee, the headache would probably go away. Okay, so now y'all know. Yeah. Tyrosine?
Yep.
What is
Before
it?
and after.
Is it,
It's like serotonin.
okay, so it's a neurotransmitter
Yeah, it helps.
sub, or is it just an analgesic? Does it just tamp down on the symptoms of the headache?
Yeah, well, it improves your mood. It stabilizes your.
But do you know the mechanism, is what I'm asking. Okay. So, so there are other drugs you can take. So what about,
5-HTP.
yeah, that's a serotonin. Excedrin. What's Excedrin got in it? Caffeine.
Yeah. Yep.
Yep. Yep. So there's something called a rebound headache. Have you heard of that? Yeah. Usually you get rebound headaches more with Excedrin than you do with aspirin. Why? It's the caffeine as well. So you have a headache, so you take a painkiller. I don't care which one you take. And then it wears off 4 to 6 hours later, and you have a headache, so what do? You take some more, and you take some more, and you take some more, and then you finally get to the point where the original cause of your headache is long gone, but now you're more or less stuck with, I need this painkiller just to keep my head from hurting. What's the cure for that? For a rebound headache? Tough it out, dude.
Deep breathing.
Yeah, well, yeah, anything you can do to reduce it, but chances are you're just going to have to live with a withdrawal from the Excedrin or whatever, if that's what the problem is. So how important is chemical communication? It's pretty darn crucial. Okay, so mechanisms of secretion. If it's a protein or another water-soluble chemical that's too big to use a channel, then chances are you're going to make it, if it's a protein, you're going to make it in the rough ER. You're going to store it in secretory vesicles, which that's the job of the Golgi apparatus to make. And then you're going to release it by that process we just learned called exocytosis. Okay, so secretion by exocytosis is the way that you handle proteins and other water-soluble secretions. And please note that calcium ions stimulate exocytosis. This is one of the things that should go on your, if you're a list maker, start a list of things calcium does for me. What's already on our list of things calcium does for you? Helps your bones. And now you know that it stimulates secretion by exocytosis. If it's a lipid substance, like our steroid hormones, you can't store them in a cell. Steroids, like cholesterol, will zip right through the lipid bilayer. They're small lipids. You can't hold them in a cell. So when you secrete a lipid-soluble hormone like testosterone or it goes into muscles, doesn't come out of muscles. So when it comes out of the gland that's secreting it, it's just zipping right through the membrane. When it gets to the muscles, it's just zipping right in. Absolutely. Okay, steroid hormones zip right through plasma membranes without even really slowing down as far as I know. So what you do with your steroids, you can't store them in the cell that makes them, but you can kind of hang on to them in the blood. And as we'll learn in AMP2, you have proteins in your plasma whose job it is just to hold on to these steroid hormones. And so you kind of load steroids into your bloodstream, and they stay in the bloodstream longer that way. But you can't store them in the cell that makes them. Any questions about this? There's a very distinct difference. It's going to matter when we get into hormones for sure. All right. There's another kind of communication besides releasing a chemical and having it bind to a receptor. Our second kind of communication is that we can have electrical signals. And we create electricity in the body by moving ions around, positive or negative ions. As they move, they create electrical current. So we can stockpile ions in a cell and then let them out when we're ready by opening a gate. Okay? So if we can stimulate the opening of a gate of a sodium channel, it's only going to let sodium flow. Sodium's positively charged, so as sodium flows, that creates electrical current, and we can use that electrical current to do work. There are 3 kinds of tissues in the body that can use electrical current to do work. Are you ready for the list? One is nerve cells. I bet you already knew that. The second one is muscle cells, and I'm going to add a third one to the list, gland cells. So nerve cells will send nerve impulses, muscle cells will contract, gland cells will secrete due to electrical signals. They also may do that based on chemical signals, but they all generate a certain electrical current. All right, so that concludes our very rapid discussion of communication. Obviously, we're going to come back and talk about that when we get to the endocrine system, when we get to the nervous system, when we get to the muscle system. We'll talk about those secretions. I want to get started on metabolism because I want to make sure I have plenty of time tomorrow to get through the rest of what we're doing. So we're going to talk briefly about breakdown and then just start on synthesis, and then we'll call it quits. All right, so we have two categories of metabolism. Metabolism is everything. Remember, it's the sum total of all chemical reactions. And there are 2 categories. Catabolism is breaking stuff down. Anabolism is building stuff up. So if I'm making bigger molecules, that's anabolic. Okay? So we use the term anabolic steroids. What does that mean? They're in general not accepted. But what are we doing with anabolic steroids?
Cutting,
And what do you mean by cutting?
like lean muscle.
So you're talking about having a cut profile where the edges of a muscle are well defined? Is that what you mean by that? Not cutting like in I have
No.
a psychosis and so I cut myself. Yeah. Okay,
Getting-
so
Getting rid of excess body fat.
getting rid of excess body fat, but also building up the muscle, right?
Yeah, yes.
Okay, so the anabolic part of an anabolic steroid is the role of anabolic steroids in causing protein manufacture in cells. It stimulates anabolism. It builds protein. Does that make sense to everybody? So catabolic is breakdown. And if you have trouble trying to remember which term means which, just think of catastrophe. Is that more of a breaking down or a building up? Definitely a breaking down. So catabolism would be catastrophic. It's breaking stuff down. You do catabolism in your digestive tract when you're digesting food. That's technically not considered metabolism. Why not? Is it inside of a cell? If it's in your stomach, it's actually technically outside your body, even though you swallowed it. It hasn't crossed the barrier into a living cell yet. So we do extracellular digestion in our digestive tract. So metabolism refers most specifically to functions inside of individual cells. Okay, so if I'm doing catabolism inside of a cell, I might be on a calorie-restricted diet and I'm trying to rely on energy from my fat reserves, and so I'm breaking down fats in my adipose deposits. That would be catabolism. If I carry that too far, I may start breaking down my muscle protein. That's still catabolism. Okay? So if you're breaking down large molecules into smaller ones, that's a catabolic process. The main reason we do that is to produce ATP, but we also do that to provide nutrients for other processes inside of a cell. Okay? The term endotherm is the last term in the left column. Do you see it?
Yes.
What does that mean? What does endo mean? Within or inside, and therm refers to heat. So we're endotherms. That means we generate our own heat. Okay? What's the opposite? Ectotherm. Can you name one?
Lizard.
Yeah, a lizard, snakes, frogs, fish for the most part. There are some fish that are endothermic. What about, what about dinosaurs? What about birds? Are birds endothermic or ectothermic? All mammals are endothermic. Let me just get that done. So dogs, cats, rats, whatever, we're all endothermic. What about birds? Anybody know? You want to take a guess? Are they sluggish in cold weather?
No?
I've never seen one be sluggish. Don't they have nice feathers to keep them warm?
No?
Endotherms. Birds are endotherms. Okay, so what about dinosaurs?
Active.
How would you know?
They're reptiles.
You can actually tell by looking at the fossils. Most fossils of dinosaurs are bones, of course. And if you look at the bone of a dinosaur fossil, it looks like a bird or a mammal bone. It's got blood channels running through it, just like ours. And recently they found fossils of dinosaurs with the imprint of feathers. Did you know that? Dinosaurs had feathers? At least a lot of them did. And the presence of feathers tells you a couple of things. One, they're insulating. The only reason you would insulate is to keep heat in, not to keep it out. So not only were dinosaurs endotherms, but birds are now classified as dinosaurs. Did you know that one? Dinosaurs are still with us today. We just call them birds. Okay, well, that's all fun. Crocodiles are the closest reptilian relative to birds, but they're still classified differently. Okay, so with our anabolism, let's talk about that. Remember, we're making things, bigger molecules out of smaller ones. We have to build our polymers. What's a polymer? What does poly mean? Many. Mer or mer means unit. So polymer is an assemblage of a lot of units. Biologists in general don't like to count past 2, I've found. Sometimes we'll count to 3 if we have to, but usually not beyond that. Occasionally 5, but that's pushing it. Okay, so a monosaccharide is 1 sugar. A disaccharide is a double sugar, 2 monosaccharides stuck together. After that, we tend to quit counting. A dipeptide is 2 amino acids stuck together. A tripeptide is 3. After that, they're either oligopeptides or polypeptides. What did oligo mean? Short, few, okay? And poly means many. So most of our functional proteins are polypeptides, and we just kind of lump them in that category because some have more than others and there's no specific numbers. Everybody okay with that idea? So in general, we use monosaccharides, disaccharides, oligosaccharides, and polysaccharides, and the oligos and polys don't have any specific set number. Is that all right with everybody? For our lipids, the main lipid category that is used in the form of a large molecule is our triglyceride. We just did those yesterday, so what's a triglyceride made of? It's one of the cases where we count to 3. Triglyceride is 3 fatty acids stuck to a glycerol. That's our fats and oils that we just talked about yesterday. These are large molecules. If you eat fat, you have to digest it into smaller things before you can absorb it. Too big to go across plasma membranes. So if you're going to store fat, you've got to build up, you've got to do anabolism of these fats in order to store them. Proteins and nucleic acids are in special categories. We'll talk about them tomorrow. Okay, but in general, the processes of anabolism require ATP. It takes calories in food to make ATP. It takes ATP to build these molecules. So in some cases, if you have a person who is deficient in their diet and it's causing a failure to thrive, whether it's a kid with cystic fibrosis or maybe a kid who's just malnourished, generally they're not getting enough protein in their diet, but sometimes they're simply not getting enough calories to make the protein that they need. So absolutely high requirement for protein. And then the term monomer, what is that? That's the 1 unit, so the 1 glucose molecule or the 1 amino acid molecule. And then we also need vitamins and minerals. Vitamins and minerals are very often used as enzyme cofactors. Vitamins and minerals, not always, but often, enzyme cofactors. And a cofactor for an enzyme is a non-protein substance that's needed for that enzyme to do its work. Okay? So that's our overview of anabolism and catabolism. We're going to look very quickly at some terms here, and if it's in all caps or bold print, then you need to at least know the definition of the term. All right? So our fuel molecules, what do you eat for fuel? Generally, it's carbs and fats. Okay? If you're eating protein, you're probably not eating the protein for fuel. Can you use protein as a fuel molecule? Sure. But your body has a priority list. Your liver will keep the protein and use it for fuel only if there's an excess. So if you eat more protein than you need, can it get converted to fat? Absolutely. So people on the Atkins diet that never lose weight, why are they not losing weight? Even though they're on that great Atkins diet where it's all protein, they can all the protein they want. As long as you're eating more calories than your body consumes, then you're going to store the excess as fat, protein or not. So generally, your monomers are going to be glucose from your carbs, fatty acids from your fats, amino acids from your proteins. If you're starving, like really true, not like I skipped lunch today and I'm starving, but if you are on a calorie-restricted diet or a starvation diet or you're in a concentration camp or you're a refugee, whatever, if you're truly starving, you actually start breaking down muscle protein. And those cellular proteins are being burned for fuel. Okay? If the process is anaerobic, what does that mean? does not directly require oxygen. So if you didn't have oxygen available, these processes would keep going on. And the primary one of these is glycolysis. Glycolysis refers to the breakdown of glucose. Where in the cell does it occur? Look at the screen to answer the question. In the cytoplasm. Okay? Each glucose has enough energy to make almost 40 ATPs. But from glycolysis alone, you get a measly 2 ATPs. So that's like taking one bite out of a cookie and throwing the rest away. Glycolysis is not very efficient, but it has the benefit of not requiring oxygen. Okay? In the absence of oxygen, your muscles can take that waste product, the rest of that cookie you just took one bite out of, and ferment it. And in the process, they produce lactate, or lactic acid. Lactate is a waste product in the muscles. It's the waste product of fermentation. It's not really a waste. That lactate can get reused in other parts of the body. The heart can use it, cells in the brain can use it, liver cells, happy to take that extra lactate. So we're not going to throw it away, but the muscle cells can't use it as long as they're anaerobic. So if you're doing anaerobic exercise, what does that mean? Are you fast or slow? Anaerobic. You're like sprinting instead of running a marathon, right? You're going fast, okay? So you are meeting your needs without having sufficient oxygen or sufficient time for oxygen to get to the muscles. That's the idea behind anaerobic exercise, okay? Anaerobic means you don't have enough oxygen to meet your needs. In the mitochondria, we have our aerobic processes. They require oxygen. You think of air when you see the term aerobic, but it's not really air. Air is only 21% oxygen. Aerobic refers specifically to the presence of oxygen. So what's the only part of a cell that uses oxygen? That would be the mitochondria. There are 2 steps to this process. You'll be pleased to know that we worry about the details of this in A&P2. So for now, it's enough to know that your aerobic processes in the mitochondria are going to generate another 36 ATPs per glucose. So we got 2 from glycolysis. Add that to 36 more that you get in the mitochondria. You can see that our reliance on oxygen makes sense. In fact, the muscles are almost the only tissue in the body that can do fermentation. So what happens to your brain if it doesn't get oxygen? Yeah, it's going to die, actually. So if you are undergoing surgery and the surgeon inadvertently clips a blood vessel, say you're having spinal cord surgery, it's not that uncommon. If he clips a blood vessel, a little tiny one, and cuts the blood supply to a part of the spinal cord, those are nerve cells in there. What happens within 2 seconds? Those cells start to die. Yep. So how long you can go without oxygen to most of us means how long can I go if I hold my breath? Well, don't you have oxygen in your blood circulating? Yeah. Okay, so, or you could be on a ventilator. But an individual cell, if it's a nerve cell, if you cut the oxygen supply to an individual nerve cell, 2 seconds, and it's gone. So the whole idea of I can go, a whole minute holding my breath, yeah, you're not depriving your brain of oxygen for that minute. You have oxygen circulating in your blood. So that's a different kind of a problem. All right, any questions about what's happening in the mitochondria? I think we'll skip this one. We're about out of time. And what I want to do is give you a quick overview of what's left for us to do. we've got to do protein synthesis. Well, we've got to do synthesis. We've got to do how cells reproduce, cell division. And we've got to do genetics, what happens when genes mutate. So that's going to be quite a bit. What I'm going to do between now and tomorrow is try to extract from this PowerPoint, whether it wants to give it to me or not, all the words, so that I can at least give you the outline for what's in this. Not the pictures, because you don't need to be printing all that. Okay? The links to the animations are already in your chapter summary. I've got this video recorded. I guess I can stop recording it now. And I'll post the recorded video, lecture